Welcome, curious minds, to another deep dive. Today we're embarking on, well, what might just be the most incredible journey imaginable. We're looking at the very beginning of human life, how a tiny, simple disk of cells transforms in just a few short weeks into something recognizably human. It's really a marvel.
It absolutely is.
And our mission for this deep dive is, well, a bit ambitious. We want to unpack early embryonic development so clearly that, you could actually explain the big picture to maybe a 10-year-old. So we'll be using lots of memorable metaphors, trying to anchor these core ideas. And for those who like the details, we'll connect those anchors to the actual nitty-gritty that shapes us. Think of it as a shortcut to understanding why this stuff is so foundational.
It's a great challenge because these early stages, they're microscopic, but they are literally the blueprint for every single part of our adult bodies. Understanding them isn't just, a cool story. It's absolutely crucial for grasping how our systems are built and how sometimes things can go wrong later on. We'll definitely connect these tiny events to the big picture of, well, you.
Absolutely, because when you really stop and think about it, building a person from just a few cells requires such mind-boggling precision coordination. Yet, as we'll find out, some of the first steps are surprisingly elegant, really simple ideas that lead to all this complexity later. Okay, let's dive in and unpack this.
Sounds good.
So we're picking up just after gastrulation, that amazing process where our flat disk of cells gets its three distinct layers, remember. The ectoderm, mesoderm, endoderm, they've got their basic identities. Now, the first major player steps onto the stage, the notochord. It's this transient, rod-like structure right down the middle. So what is this thing? What's its job? And why is it so important if it's, temporary?
Right, the notochord. It's absolutely pivotal. Its genius is kind of in its dual role. You can think of it as the very first pencil line drawn on a blank piece of paper, drawn right down the middle where the back will be. And it's not just sitting there, it's active. It's like the embryo's first boss, sending out signals. It forms from a specific part of that middle layer, the axial mesoderm, starting around day 16, and it's fully formed by about week 4.
Okay, day 16 to week 4.
Yeah. Its main job, defining the embryo's central axis. This dictates the entire orientation of your future vertebral column. It's even essential for setting up left-right asymmetry in your body, making sure your heart ends up on the left, that sort of thing. It really kicks things off.
So it's not just a line, it's like a director telling everyone where to go.
Exactly, a powerful conductor. And what's really fascinating is that while most of it disappears, it is transient. One part actually sticks around in adults. It becomes the nucleus pulposus. That's the jelly-like center inside your intervertebral discs. Gives your spine flexibility.
No way. So that first pencil line becomes part of our spine forever.
That's right, permanent flexible part. But the notochord story doesn't stop there. As soon as it forms, this powerful inducer immediately sets the stage for the next huge step, building your nervous system.
Wow, okay. A temporary structure with a lasting legacy. So it's an inducer. What does that mean for building our nervous system?
Right, this leads us straight into neurulation. This is the process of forming the neural tube, which is basically the foundation of your entire central nervous system, your brain, and your spinal cord. So under the notochord's influence, these chemical signals it sends out, the overlying surface ectoderm, that top layer of cells, starts to thicken and flatten out. It forms the neural plate.
So the notochord is basically shouting up at the cells above it, hey, you guys, you're the neural plate now.
Pretty much, yeah. Imagine taking a flat sheet of paper that's your neural plate. Then, around day 18, it doesn't just stay flat. It starts to fold inward, right down its central axis. That creates the neural groove, like you're pushing down the middle of that paper. As that happens, the sides, the lateral edges of that groove start to lift up. Those are the neural folds. Now picture those folds moving towards each other, closer and closer, until they touch and fuse together in the middle. Like a zipper. Closing up right down the middle of the embryo. That's the perfect metaphor. It's exactly like a zipper closing, starting kind of in the middle, the future neck region, and zipping up towards the head and down towards the tail simultaneously. This fusion turns that flat plate into a hollow neural tube, and that tube is the beginning of your entire central nervous system. This zippering process is super critical and happens on a really tight schedule. The anterior neural pore, that's the head end of the zipper, closes around day 25, and the posterior neural pore, the tail end, closes around day 28. Once that bottom end closes, neurulation is complete. You've got a rudimentary spinal cord and the very beginnings of your brain.
Day 25 and day 28. That's incredibly precise for something so fundamental. And while this zipper is closing, you mentioned there's sort of a side story, some cells making a great escape. Tell us about these neural crest cells.
Ah, yes. The neural crest cells. They are truly amazing cells. As those neural folds are coming together and fusing to form the tube, some masses of ectodermal cells right on the outer edges, the lateral side, actually detach themselves. They break free. These are the neural crest cells. They're also transient, but incredibly important. They're famous for being highly migratory. They travel all over the developing embryo, and they are remarkably multipotent. That means they can turn into a huge variety of different cell types, parts of your peripheral nerves, pigment cells in your skin, even some bones in your face and skull. They're like the embryo's utility players.
Wow, nature's shapeshifters. That's fascinating. But, okay, back to the zipper. What happens if it doesn't close properly? If that fusion isn't complete?
Yeah, that's a critical point. When the neural tube fails to close completely, we get what are called neural tube defects, or NTDs, for instance, anencephaly. This happens if the failure to close is up in the cranial, the head region. Sadly, this means most of the brain fails to form. Then there's spina bifida. This is when the tube fails to close somewhere lower down, anywhere from the neck region caudally towards the tail. It's most common in the lumbosacral area, the lower back. And this is where understanding this connects directly to prevention. A really significant number, maybe 50 to 70% of all NTDs can actually be prevented by adequate daily intake of folic acid, B vitamin, before conception and during early pregnancy.
That's incredible. Just understanding this early step leads to a direct way to prevent serious conditions. That's a powerful takeaway. Okay, so we've established the central axis with the notochord and the nervous system starting with the neural tube. What about the sides? What's happening alongside all this central action?
Good question. Flanking the neural tube, this is where the somites come in. During that third week, the paraxial mesoderm, remember, part of that middle layer running alongside the tube, it doesn't stay as a continuous band. It starts to organize itself. First into segments called somitomeres, and these then condense into distinct blocks called somites on either side of the neural tube. The best way to think of them is like Lego bricks or building blocks lining up neatly on both sides of that central neural tube structure.
Okay, Lego bricks. Little segmented blocks just appearing, almost like pre-vertebrae.
Exactly like that. And they appear in a very specific sequence from head to tail craniocaudally. About 3 pairs form each day. So by the end of week 5, you've got something like 42 to 44 pairs lined up. Like the Lego bricks, they are transient structures in their original form, but they are absolutely essential. They establish the fundamental segmented body plan of vertebrates. They're crucial for forming your vertebrae, your ribs, your muscles, and even guiding nerves.
So these Lego bricks, they all look the same at first, right? But clearly they don't all end up as the same thing. How do they specialize? How does each block know what it's supposed to become?
Right, they undergo differentiation. Each somite splits up, essentially, into 3 key parts with different fates. First, there's the sclerotome. This is the part closest to the neural tube and notochord, sort of the ventromedial part. Think of these cells as the bone builders. They migrate and become cartilage cells, chondrocytes, which will form your vertebrae and ribs. They build the axial skeleton, the skull base, vertebral column, ribs.
Okay, so sclerotome equals bone builders for the backbone and ribs.
Got it. Precisely. Then the remaining part is called the dermatome. It's sort of a double-layered structure on the more dorsal lateral side. This then subdivides further. The dermatome comes from the central region of the upper dorsal layer. Think of this as the skin patch. It generates the dermis, the deep layer of skin, specifically for the back. And then the other part is the myotome, which is mostly the lower layer. Its cells become myoblasts, the precursors to muscle cells. These are the muscle makers. They give rise to all the skeletal muscles of your trunk and your limbs.
Wow, so each Lego brick splits into bone builders, a skin patch for the back, and muscle makers. That's incredible organization. So this early segmentation, how does this blueprint laid down in week 3 or 4 actually affect, what a doctor sees or tests in an adult patient?
It has a huge and direct clinical impact. This relates to somite innervation and the concepts of clinical dermatomes and myotomes. See, the somites actually dictate how the spinal nerve axons grow out, and the sclerotomes do something clever. They resegment. Each sclerotome block splits into a top half cranial and a bottom half caudal. Then the bottom half of one fuses with the top half of the one below it to form a single vertebra.
Hold on, why do they do that split and fuse thing?
It's a brilliant trick, because the spinal nerves are growing out towards the myotomes before this resegmentation happens. By splitting and reforming, the vertebrae end up being offset from the original somite block. This ensures the spinal nerves exit between the vertebrae, giving them a safe passage out from the spinal cord.
That is clever. Okay, so the nerves get threaded through perfectly.
Exactly. And here's the key point for clinicians. Each myotome, muscle group in each dermatome, skin area, retains the nerve supply from its original segment of origin. So the pattern of innervation in your adult body directly reflects that early embryonic segmentation set up by the somites. Clinically, a dermatome is that specific area of skin where sensory information comes from a single spinal nerve root. You have about 30 of them mapping out the body. A myotome refers to the group of muscles primarily controlled by the motor fibers from a single spinal nerve root.
And why is knowing that map so crucial?
Because it's essentially the body's original electrical wiring diagram. It's laid down precisely in the embryo and stays consistent throughout life. So testing sensation in dermatomes and muscle strength in myotomes is fundamental in a neurological exam. If someone has symptoms like pain, numbness, or weakness, maybe from a condition like radiculopathy that's an irritated nerve root, often from a disc problem mapping those symptoms to specific dermatomes, and myotomes can tell the doctor exactly which spinal nerve root, and therefore which level in the spine, is likely affected.
It lets you trace the problem right back to the source. That makes perfect sense. Wow. Okay, so we've got our central axis, our developing nervous system, our segmented Lego bricks building bone, muscle, and skin on the sides. But hang on, isn't the embryo still basically a flat disc at this point?
You're exactly right. We've built the key components, but the overall shape is still quite flat, like a three-layered pancake. This sets the stage for the next really dramatic transformation, embryo folding. This happens pretty quickly, mainly between day 24 and day 28. This is where that flat, trilaminar disc morphs into a cylindrical-shaped embryo. It establishes that fundamental tube body plan. You can imagine taking a flat blanket and wrapping it around yourself to make a tube, or maybe like a deflated balloon that's flat, and then you inflate it and it becomes more 3D, more cylindrical.
So it's not just growing bigger, it's actively folding and tucking and rolling into a 3D shape.
Precisely. It's a very active process. And this folding, or flexion, also starts to pinch the embryo off from the surrounding membranes, like the yolk sac. Eventually, it's only attached by the developing umbilical cord. And the folding happens in two main directions at the same time. There's longitudinal folding, head to tail, or cephalocaudal flexion. This creates the head fold and the tail fold. And simultaneously, there's transversal folding or lateral folding. The two sides fold downwards and inwards towards the midline.
Okay, so it tucks in from the top and bottom and rolls in from the sides all at once. That sounds complicated. What are the big results of all this folding?
The consequences are huge, totally reshaping the embryo. For example, that cranial or head fold tucks the top rim way down ventrally towards the front. This movement actually forms the ventral or front surface of the future face, neck, and chest. And crucially, it swings the developing heart tube down from near the head into its proper position in the future thorax. At the same time, as the sides fold in, the edges of the ectoderm sheet meet and fuse along the ventral midline. This completely wraps the embryo in skin. It becomes a continuous outer covering, and the same thing happens internally. The edges of the endodermal layer also meet and fuse, forming the inner gut tube.
Ah, so the folding literally creates our basic body plan, a tube within a tube.
Exactly. That's the classic description.
Okay.
A tube within a tube body plan. You have the outer ectodermal tube, which is the skin, and the inner endodermal tube, which is the primitive gut. And the space between those two tubes is primarily filled with mesoderm. Also, during folding, a structure called the septum transversum, which initially lies quite cranially, gets repositioned down into the torso. It's important because it will contribute significantly to forming the diaphragm, separating the chest and abdomen.
That's amazing. Folding creates the whole layout. So you mentioned the space between the outer skin tube and the inner gut tube. What happens in that space? What does it become?
Right, that space, lined by mesoderm, is where the intraembryonic coelom, or the main body cavity, forms. To get there, we have to rewind just slightly, back to the third week again. The lateral plate mesoderm, which is the mesoderm furthest out to the sides, actually splits into two distinct layers before folding really gets going. There's a dorsal layer closer to the back called the somatic mesoderm, or somatopleur, which stays associated with the ectoderm, and a ventral layer closer to the belly called the splanchnic mesoderm, or splanchnopleur, which associates with the endoderm gut tube. As the embryo undergoes that dramatic lateral folding, these two flaps of mesoderm, with the space between them, fold down towards the front midline. They meet and they merge.
So they started flat, split apart, and then the folding brings the edges of that split space together to enclose it.
Exactly. That enclosed space is the intraembryonic coelom. It's the primitive body cavity. Think of it like a builder creating the main internal space within the house of the body. And this initial single cavity then gets partitioned. It gets subdivided, like creating different rooms by membranes and structures like the septum transversum we mentioned. These rooms become the major body cavities, in the adult. The pericardial cavity around the heart, the two pleural cavities around the lungs, and the large peritoneal cavity in the abdomen and pelvis, where most of your digestive organs reside.
Wow. It's like internal architecture being built right there from that initial split and fold. It really is incredible how much gets established, how much of our fundamental structure is laid down in just these first few weeks.
It truly is astounding, isn't it, when you just pause and think that every single one of us started as that simple flat disc
Hmm.
and then went through these incredibly precise, almost choreographed steps. Axis formation, tube formation, segmentation, folding. It's so efficient, so precise. And these microscopic events, which are easy to just gloss over, they really dictate the fundamental rules for our entire adult anatomy and even, as we saw with NTDs, our susceptibility to certain conditions.
It definitely makes you appreciate the intricate dance of cells and signals needed, a reminder that we all began as this incredibly detailed, rapidly changing blueprint.
And, this raises an important question, especially for anyone listening who's heading into medicine or healthcare. How does really understanding these building blocks and these folding rules of the embryo, how does that fundamentally change how you might approach diagnosing a problem or even thinking about treatment in an adult patient years later?
That's a powerful thought to end on, knowing the origin to understand the present. Well, thank you for taking us through that incredible journey, and thank you all for joining us for this deep dive into the very beginnings of human life. We hope you enjoyed tracing your own amazing origin story.